Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF Entrance Exam question paper conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. I have also uploaded still previous year's question papers of DBT BET JRF. You can see in my channel playlist. To the first question. What is the relationship between doubling time and specific growth rate of a microbial culture? So doubling time, that is G, it is given by the equation 0.6931 by mu, where mu is the specific growth rate. So you can see from the equation G is inversely proportional to mu. So the answer here is option 2. Doubling time is inversely proportional to the specific growth rate. Answer is option 2. Then next one. Given below are two statements. One is labeled as assertion and the other is labeled as reason. All expression vectors are also cloning vectors. That's correct. Expression vectors contain the features of the cloning vectors. Yes. They have multiple cloning site, origin of replication. So all these features like those of cloning vectors can be seen in expression vectors also. So that is correct. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. An athlete participating in an early morning marathon before breakfast is likely to derive most of the energy for muscles from. So here the athlete lacks carbohydrates because he doesn't have his breakfast. So your body or his body uses more fat as energy. So the answer here is option 2, fats. Then next one. Which one of the following techniques will you resolve proteins only on the basis of their molecular weight? So only on the basis of molecular weight is SDS page. Native page separates protein based on size, charge and shape. So the answer here is option 2, SDS page. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion. Spectrophotometry is a technique used for quantitative estimation of biomolecules in solution. Spectrophotometry is based on the Bragg's law. So first statement, that spectrometry is a technique used for quantitative estimation of biomolecules in solution, is correct. You estimate various biomolecules like proteins, carbohydrates, DNA, RNA, etc. by spectrophotometry. But it is not based on Bragg's law, but it is based on Beer-Lambert law. So A is correct, but R is not correct. Answer is option 3. Then next one. Which one of the following techniques is used for detecting protein-protein interactions in vivo? So here they have given various options. Surface plasmon resonance, it is also used for protein-protein interaction but not in vivo. Then ELISA, it detects the counts of certain antibodies, antigens, proteins in a given sample. Then yeast 2 hybrid assay, it is used for protein-protein interactions in vivo. Yeast 1 hybrid assay is for DNA and protein interactions. So the answer here is option 3. Then next one. What is the purpose of quantitative real-time PCR? It is used to quantify gene expression levels based on RNA transcript. Answer is option 2. Then, which one of the following method is based on the binding of Quamacy brilliant blue to proteins to determine the protein concentration? It is Bradford's method. Answer is option 3. Then next one. Match the items in list 1 and items in list 2. Surface plasmon resonance is protein-protein interaction. So A4, so 2 and 4 are not correct. Options 2 and 4 are not correct. Isoelectric focusing, it is for resolving protein mixtures based on their isoelectric point. So B3. Single strand conformation polymorphism. It is based on the fact that single stranded DNA has a defined conformation. But when there is alteration in the conformation of the single stranded DNA by a change in base sequence, or sorry, a change in a single base, then then this can cause the single stranded DNA to migrate differentially under non-denaturing conditions. So single strand conformation polymorphism is conformation difference of DNA. Conformation difference of DNA. Answer C1. Then next one, denaturing gradient gel electrophoresis. So here they separate DNA strands based on GC-AT base pairs. So during this process, what they do is they expose the DNA to a gradient of denaturant at high temperatures within a polyacrylamide gel. Then as the DNA sample, it progresses through the gel from low to higher denaturant concentration. It begins to melt at varying points. So here if GC is triple bonded and AT is double bonded, so GC will separate at different denaturing conditions and AT will separate at different denaturing conditions, and depending on the amount of GC and AT also separation will be different, and it is used to detect DNA mutation and heteroduplex analysis. So the answer here is option 1, A4, B3, C1, D2. Then next one, how much calcium chloride is required to make 40 ml of 0.02 molar solution? Assume molecular weight of calcium chloride is 219. So for molarity you have got a formula 1000 Wb by Mb into V, where M is molarity, Wb is weight of the solute required, Mb is the molecular weight of the solute, V is the volume of the solution. Now substitute and calculate for Wb. You will get the answer as 0.1752 grams. Answer is option 3. Then next one, given below are two statements. Statement 1, the Michaelis constant characterizes the affinity of an enzyme to its substrate. That is correct. Higher the value of the Michaelis constant, stronger is the binding of the enzyme to the substrate. That is wrong. The value of Km, it is inversely related to the affinity of the enzyme for its substrate. So higher Km value, it corresponds to lower binding affinity, low enzyme affinity for the substrate. So the answer here is option 3. Statement 1 is correct, but statement 2 is incorrect. Then next one, the first commercially produced plant secondary metabolite using plant suspension culture in bioreactor was. So it was chiconin. Answer is option 1. Then next one, two proteins have approximately the same molecular weight and the same isoelectric point. The best way to resolve them would be using reverse phase chromatography. Why, I will tell you. See the other options. Gel filtration, it is based on difference in molecular size of protein, but here they have the same molecular weight, so gel filtration cannot be used. Isoelectric focusing separate protein based on their different isoelectric points. Here the same isoelectric point, so that also cannot be used. Then TLC is used to separate mixtures based on their differences in polarity. They have not mentioned anything regarding polarity here. The polar amino acids are retained on the stationary phase. So that also is not possible. Then reverse phase chromatography. It separates molecule based on hydrophobic interactions. Reverse phase chromatography. So naturally in these proteins there will be hydrophobic amino acids also. So they can be these proteins which show hydrophobic interaction. They will bind to the hydrophobic ligands which are attached to the stationary phase in reverse phase chromatography, and then they can be eluted out. So the answer here is option 1. Then next one, For an enzyme following Michaelis-Menten kinetics, the catalytic efficiency of the enzyme is measured by Kcat by Km. Then, in alcoholic fermentation, carbon dioxide is evolved during. So, during alcoholic fermentation, glucose undergoes glycolysis and produces pyruvic acid. Pyruvic acid, it is a 3-carbon molecule and it is converted to acetaldehyde by losing 1 carbon dioxide molecule via decarboxylation. So the answer here is both decarboxylation of pyruvic acid and formation of acetaldehyde. Answer is option 3. During the life cycle of microbes, at which stage do they produce primary metabolites? So it is during the exponential phase. Answer is option 2. Then next one. Under high concentration of glucose, ethanol production by yeast cells instead of increasing cell mass via TCA cycle is described as. So there is high concentration of glucose. Ethanol is produced by yeast cells instead of increasing cell mass via TCA cycle. They go for ethanol production. So what is this called? It is called as Crabtree effect. Raman effect, Simpson effect is not correct answer. Warburg effect. Warburg effect, it is a phenomenon observed in cancer cells wherein cells ferment glucose to lactic acid using glycolysis even in the presence of oxygen. That is called as Warburg effect. Then next one. Which one of the following statement is correct? The genome size and the number of genes are directly proportional. No, they are not at all directly proportional across species. So the answer here is option 4. The genome size is not directly proportional to the number of genes across species. Answer is option 4. The next one. What is the mutation rate per generation for humans? It is 1.3 into 10 raised to minus 8 per base pair. Then, in nature, 20 different amino acids can be coded by 4 different nucleotides A, T, G and C. Suppose the number of available nucleotides increases to 6, A, T, G, C, X and Y. For the genetic code to be made up of codons having equal number of nucleotides, what would be the minimum number of nucleotides required for each codon? Now you have learned that 4 raised to 3, that is 64 codons code for 20 amino acids and 4 raised to 3. So it shows it is a triplet codon. So here it is 6 raised to 2. It will give you 36 and 36 is sufficient for coding 20 amino acids. So it will be a doublet codon. Answer here is option 3. 2. Then next one. Match the items in list 1 with items in list 2. 1000 genomes. 1000 genomes is catalog of genomic variants. GenBank, it is a nucleic acid sequence database. OMIM, it is for genes and disease phenotype database, and UK Biobank, it is a biomedical resource with genetic, environmental and clinical data. Answer here is option 2. Then next one. The following sequence of DNA seems to form a structure. When the third base is changed from C to G, the DNA loses its structure. However, if the background of this change, the 15th base, which is the third last one, is changed from G to C, the DNA regain back its structure. The most plausible reason for the observation is, so this is your DNA sequence. I have rewritten here. There are 17 nucleotides. This is how it forms stem loop structure. A with T, T with A, C with G. So here if you see, this is what changes they have made to third and 15th. So third base is changed from C to G. Here it was C first, now it has changed to G, and 15th base is changed from G to C. G to C. So it will regain its stem loop structure back. So the answer here is the third base and the 15th base are paired in stem-like structure. Third and 15th base. Third and 15th base are part of loop-like structure. Now loop-like structure is this one. I didn't have much space to draw properly, that's why it's looking like this, but still I have made it. GAATT is the loop-like structure. The third and 15th bases have steric clashes. No, the third base and the 5th base and the 15th base with the 13th one. That is also not correct. Third base pairs with the 15th base. So the answer here is option 1. Then next one. Unfolded protein response occurs when the cells are stressed. Unfolded protein response is a hallmark response by the nucleus to protect the genome. Choose the correct answer from the options given below. So unfolded protein response occurs in a cellular stress, and it is related to the endoplasmic reticulum. And this unfolded protein response is activated in response to accumulation of unfolded or misfolded proteins in the lumen of the endoplasmic reticulum. So here they have given it as the response by the nucleus. It is not nucleus, it is endoplasmic reticulum. So statement 2 is incorrect, whereas statement 1 is correct. So the answer here is option 3. Statement 1 is correct, but statement 2 is incorrect. Then next one. While analyzing a multiple sequence alignment of homologous protease sequences, the following was observed. So protease is an enzyme. So they have done multiple sequence alignment of homologous protease sequences. The 14th position was variable but always encoded a hydrophobic amino acid. The 17th position was conserved and was serine in all the homologous sequences. The 29th position was variable but always encoded by an aspartate or a glutamate. The 50th position was conserved and always encoded by a phenylalanine. Which statements do you think are consistent with the above observation? Residue 14th is buried amino acid. That is correct because it is a hydrophobic amino acid, so naturally it will be in the interior of the protein. So that is correct. A is correct. 17th position may be part of the active site of the protein. Yes, that is also correct because it is conserved and it is always a serine there in all the homologous protease sequences. So it could be a active site of the protein. That's also correct. 29th residue is a buried amino acid. 29th residue is aspartate and glutamate. So they are polar amino acids, aspartate and glutamate, so they need not be buried. So 29th is so not correct. Then 50th position is an exposed amino acid. 50th position was conserved and always encoded by phenylalanine. Phenylalanine is a non-polar amino acid, so it will not be exposed amino acid, it will be buried. So the correct answer here is option A and B only. Then next one. The delta G of unfolding reaction of a monomeric protein, folded to unfolded, varies with the concentration of guanidine hydrochloride with the following relationship. So delta G is equal to M into GDNHC, guanidine hydrochloride plus 10 kilocalories per mole, where M is minus 2 kilocalories per mole per molar. What is the concentration of guanidine hydrochloride at which half of the protein is unfolded? So they have given the equation here, delta G is equal to M into guanidine hydrochloride plus 10 kilocalories per mole. Now substitute the value of M there. So it will be minus 2 into concentration of guanidine hydrochloride plus 10. The delta G at which half of the reaction is unfolded is 0. Substitute the value of delta G. Then calculate concentration of guanidine hydrochloride. So 10 by 2 it is 5 molar. So the answer here is option 3, that is 5 molar. Then next one. A protein forms dimer XX giving X plus X. The Kd, that is dissociation constant of the reaction, is 1 micromolar. The 1 micromolar concentration of monomer X, what is the concentration of dimer X? So dissociation constant, it is given by concentration of monomer divided by concentration of the dimer. Now substitute the values. I have converted micromolar into molar, multiplying by 10 raised to minus 6. So concentration of dimer will be 10 raised to minus 6 or 1 micromolar. Answer is option 2. Now next one. Sometimes phi and psi angles can be used to construct the 3D structure of a protein. Which one of the following statements are true with respect to the process? The omega angle is required to model the final 3D structure. That is correct. Omega, psi and phi angles, all three are required to model the final 3D structure of the protein. Phi and psi are not sufficient to model 3D position of the side chain atoms. That is also correct. Then the allowed regions of Ramachandran map is sufficient to model the 3D structure ab initio. It is Ramachandran map is used for 2D structure, not 3D structure. So C is not correct. Phi and psi are sufficient to fix all main chain atoms except for glycine, which is achiral. Not only phi and psi, you require omega also. So D is also not correct. So answer here is option 1, A and B only. Now next one. Which of the following forward primers, they have asked only for forward primers, will you use to amplify the DNA sequence given below? So this is the DNA sequence. I have separated the DNA strands. This is what they have given. This is the where the forward primer is. Here will be your reverse primer in this direction. So they have asked only forward primer. So it is ATGC CAA. That is option 1 is your answer. Then next one. Approximately how many helical turns are generally present in a 4800 base pair long non-supercoiled bDNA? So number of helical turns in a DNA molecule is length of the DNA in base pairs divided by number of base pairs per helical turn. So for bDNA we know number of base pairs per helical turn is 10. And length of the DNA they have given it as 4800 base pair. So divide by 10, that will give you 480 turns. Answer is option 2. Then next one. Which one of the following is formed when cysteine base is deaminated? So when cytosine is deaminated, you get uracil. After solving a protein structure by X-ray crystallography, you found that a few residues are in the disallowed region of the Ramachandran map. Which one of the following are plausible explanation for this observation? There may be errors in the structure of these residues. That could be an possible explanation. There are many side chain interaction in those residues that offset the disallowed cost. That's also correct. The proteins may have lot of proline residues. No. The protein may not be in a proper folded state when it was crystallized. That is also not correct. So the answer here is A and B only. Answer is option 1.